Hello, I'm Haila Schultz, and I'm a graduate student at the University of Washington. Today I'll be discussing a project in collaboration with NOAA applying metabarcoding to investigate zooplankton and ichthyoplankton diversity in the Salish Sea. So zooplankton comprise an important link in marine food webs and are incredibly diverse. Responses of individual zooplankton species to chemical and physical conditions vary. However, the early life stages of plankton taxa often look similar, making it impossible to separate many of them via microscope identification. This limits our ability to get an accurate read on true species diversity and community composition. It also limits our understanding of the environmental conditions that structure these communities. Omics methods are arising as a way to address this issue. Compared to microscope identification, DNA metabarcoding is faster and allows for more complete and accurate IDs, even for cryptic taxa and early life stages. Metabarcoding allows us to perform barcoding on bulk zooplankton samples, processing millions of sequences for many species. We use multiple general and targeted assays for multiple nucleic and mitochondrial genes in conjunction with custom bioinformatics pipelines. We collected netto samples over 4 years from 7 long-term monitoring locations in the Salish Sea in Washington State. This estuary is home to a wide array of environments with varying water chemistry, allowing us to address two main research questions. Number one, how do zooplankton communities differ across locations, seasonal and annual patterns, and water chemistry? And number two, how are these communities responding to changing environmental parameters, notably ocean acidification, hypoxia, warming, and invasive species? This figure shows results from some of our samples from April and September 2018. The 7 stations are located on the x-axis, and the relative abundance of reads of 19 common species are located on the y-axis. As you can see, zooplankton community composition and diversity clearly vary by station and season, reflecting environmental differences. Going forward, we will continue to refine our assays in ground truth with microscope taxonomy, taking a first step toward a broader assessment of the relationships between zooplankton and ocean chemistry. Metabarcoding has great potential for use in monitoring programs to assess the diversity of plankton communities in conjunction with ocean acidification, hypoxia, and global temperature rise. In the future, this method can provide us with relatively prompt feedback on zooplankton communities, allowing us to better understand how they respond to our changing oceans. Thank you.